So if we bring it back to the big picture, here's what we've got. So we've just talked about glycolysis, and again, one of the major functions of glycolysis is to break down a glucose molecule so that high energy electrons are released. Those high energy electrons are picked up by NAD to become NADH. That's the molecule that then delivers the electron to the electron chain where ATP, a whole bunch of ATP, can be produced. Remember that one of the leftovers of glycolysis, this pyruvate, can have even more electrons squeezed out of it during the citric acid cycle. But it turns out that the pyruvates can't directly enter the citric acid cycle. First, it has to be converted into something called acetyl-CoA. And so what we'll take a look at next is that reaction that converts pyruvate to acetyl-CoA. And here it is. And so basically what occurs during that conversion is that this CO2 portion of the pyruvate ends up being removed. So it ends up leaving. And so again, this is the source, one of the sources of the CO2 that you end up breathing out when you're doing respiration. That CO2 that's removed is replaced by this group, so this sulfur coenzyme A group. It turns out that during this step, this conversion of pyruvate to acetyl-CoA, this is another step where high energy electrons are released, and so here's another place where NAD+ can come in and pick up electrons and a proton to become NADH. And so here's another source of the NADH that can go to the electron transport chain. So again, this pyruvate that was produced during glycolysis can enter the citric acid cycle to release even more electrons, but before it can enter the citric acid cycle, it's first converted to acetyl-CoA. What we'll do next then is take a look at what happens during the citric acid cycle. And so here is the citric acid cycle, which is actually also referred to as the Krebs cycle after the guy that discovered how it goes. And so you'll probably hear me calling it the Krebs cycle every now and then. Here's the acetyl-CoA that we just talked about. And so the point at which it enters the citric acid cycle, what happens to it is that this acetyl-CoA, which has 2 carbons in it, combines with this molecule that has 4 carbons in it in order to form this molecule that has 6 carbons in it. And so this molecule is called a citrate, or a citric acid, and so that's the reason why this whole process is called the citric acid cycle. So basically what happens during the rest of the cycle is that carbons are removed from the molecule and they leave in the form of CO2s. Some rearrangements then occur to the molecule after the carbons are removed from it. And during this whole process of carbon removal and rearrangements, these really important molecules of NADH, ATP, FADH, and GTP are produced. So we've talked about what NADH and FADH does, and we talked about why ATP is important. GTP is really similar in structure to ATP. So this guanine triphosphate is also considered to be a high energy molecule that can be used to power certain activities of the cell. So depending on the type of cell it is, it'll either produce GTP during the Krebs cycle or ATP during the Krebs cycle. We'll see actually in class later on that the citric acid cycle is important for another reason other than making these NADHs and ATPs, and so again we'll save that for class, but for now what we're trying to hammer home are these main points about the citric acid cycle, so we'll just list them. So things to note about the citric acid cycle is one of its main functions is to squeeze even more energy out of the pyruvates that are produced at the end of glycolysis. And so remember that pyruvate first has to be converted to acetyl-CoA before it enters, where the energy can be squeezed out of it. And so by energy, we're referring to these things: NADH, FADH. Those are molecules that contain high energy electrons that can be used in the electron transport chain. And we've already talked about why ATP and similarly GTP has high energy. Right? So that occurs during the citric acid cycle. Another thing to note about the citric acid cycle is that it basically involves different 4 to 6 carbon carboxylic acids, and they're carboxylic acids with multiple carboxyl groups that are converted into one another. And so what we're saying is that all of these molecules that are produced during the citric acid cycle, they all have anywhere from 4 to 6 carbons in them, and they all have multiple carboxyl groups. And so if you don't remember carboxyl group, here's what a carboxyl group looks like. So carboxyl groups have both a carbonyl group in it and a hydroxyl group in it. So that's kind of where the term carboxyl comes from. It has carbonyl and hydroxyl. So if you smush those two terms together, you get carboxyl. And so if you were to write it out using a chemical formula, that it's written out as COOH. It turns out that this form is easily ionized in solution, so that hydrogen can end up leaving. So this is also considered to be a carboxyl group, just without that hydrogen. So COO minus is also indicative of a carboxyl group. And so again, if you take a look at all of these molecules that are produced during the citric acid cycle, they all have multiple carboxyl groups on them. And then one other thing to note about the citric acid cycle is that it produces CO2. So again, whenever you see a step that involves removal of a carbon, so here you have a 6-carbon molecule, here you only have a 5-carbon molecule. That tells you that one of the carbons is removed in the form of a CO2. So that's the story on the citric acid cycle. And so again, bringing it all back to the big picture, we mentioned that during respiration, most of the ATPs are made during this electron transport chain. This electron chain gets its electrons during glycolysis, or from glycolysis and from the citric acid cycle, because during these two processes, high energy electrons can be released. Those electrons are picked up by a molecule of NAD+, and so after they've picked up the electron, they become NADH. Same thing here. During the citric acid cycle, the high energy electrons that are released can be picked up by an NAD+ molecule to become NADH. Remember also that the citric acid cycle produces these FADH molecules that also serve to deliver electrons. What these NADH molecules do is that, again, they deliver the electron to the electron transport chain so that a whole bunch of ATPs can be produced. Other things that are required by glycolysis, remember that there's a step where ADP, or actually a couple of steps, where ADP comes in to pick up a phosphate group to become ATP. It turns out that in order to make ADP, an AMP molecule is made. So adenosine monophosphate contains only one phosphate group. It has another phosphate group added to it to become ADP, adenosine diphosphate, and then that has then a third phosphate group added to it to become adenosine triphosphate, or ATP. Same thing here. There's a couple of steps, or there's a step during the citric acid cycle where ATP is produced. It's produced when AMP has another phosphate added to it, and then when ADP has a third phosphate added to it, then it becomes the high energy, the really high energy ATP molecule. The thing that we'll mention next is that it turns out that this whole process can be stopped, or at least slowed down, under different circumstances. So you might be thinking, well, this all seems really important, making ATPs and NADHs. Why would a cell ever want to slow it down or stop it? So if you think of it in terms of like someone who's responsible for making soup. So say that you have someone who makes soup for people, and when they're done making this soup in the kitchen, they end up putting the bowls of soup on a counter in order for people to pick it up. So if you have this soup maker that's just kind of really heavily concentrated, heavily focused on just making bowl after bowl of soup, eventually there might come a point where the soup maker will look up and see that the counter is just full of bowls of soup. And it's full of bowls of soup because nobody has picked them up. Right? So when people have stopped picking up bowls of soup from the kitchen, it probably indicates that everyone's already full. Right? So that nobody wants any more soup. So if you're the soup maker and you see that nobody really wants any soup anymore, and the proof of that is that there's a whole bunch of bowls of soup that have kind of piled up on the counter, then from your point of view, it's probably a good idea to stop making soup, right? Because it would kind of be a waste of your efforts to keep making soup if nobody's going to eat it, especially if the soup doesn't really keep for that long. So if it sits on the counter for any short period of time, it ends up spoiling. So you're really wasting your energy at that point. So the same thing kind of happens with cells. So there'll come a point where maybe glycolysis and the citric acid cycle and the electron transport chain has produced enough ATP for the cell to use. And so whenever the cell starts seeing ATPs just start piling up, that would probably be a good time then to shut down or at least slow down glycolysis and the Krebs cycle, because otherwise you're just running glycolysis and the Krebs cycle for no reason when the cell already has enough ATPs and it knows what to do with. So again, bring it back to the point. We're saying here that there are instances when the cell might be interested in slowing down glycolysis and the citric acid cycle, and that's actually what happens. So it is possible for the cell to do just that. So glycolysis and the citric acid cycle can be regulated. In other words, it can be turned on and off. It gets turned on if there's a lot of products available. So if a lot of products accumulate, then the glycolysis and the citric acid cycle shut down, or they at least slow down. And so by products, what we mean is this. So take a look at what's being made during this process. One of the major products is ATP. Right, so that's produced by the whole process. NADH is also produced. So again, we're saying that if things like NADH and ATP start piling up, then it would be a good time or a good reason to slow down this whole process. Kind of on the flip side of the coin, the thing that ends up turning on glycolysis or speeding up glycolysis in the citric acid cycle is if there's a lot of reactants available. So that's, again, a good time to turn it on. So by the reactants, so take a look at what all is needed in order for glycolysis and the citric acid cycle to occur. So you see here that ADP is required for glycolysis to happen. NAD is required in order for glycolysis to happen. The same thing with the citric acid cycle, right? So those are the reactants as well for the citric acid cycle, because NAD is required for it to occur. ADP is also required for it to occur. And so what we're saying here is that if the cell finds itself with a lot of ADPs sitting around, or a lot of NADs sitting around, that's a good time to kind of turn on or speed up this whole process. And so here's why that kind of makes sense. If you have a whole bunch of ADPs sitting around, for example, that's kind of an indicator that the cell has used up a lot of ATP, right? Because whenever a cell uses ATP, it removes one of the phosphate groups from it. So that releases a lot of energy that the cell uses to power a certain activity, and then it leaves behind an ADP molecule. So again, ADPs are indicators that ATPs have been used up. So if there's a lot of ADPs around, it's kind of a sign that the cell is short on ATP, right? It's a sign that the cell has used up a lot of ATPs and would probably like more. Same thing with NAD, right? So if there's a lot of NADs suddenly accumulated inside of the cell, it's a sign that the cell is kind of maybe starving for NADHs. Because whenever a cell uses NADH, right, it ends up dropping off its H to an electron to the electron transport chain to leave behind NAD+. So if there's a lot of NAD pluses inside of a cell, that's an indicator that a lot of NADHs have been used up, and it's probably a good time then to start making more NADHs. So that's what we mean by if a lot of reactants accumulate, it's a good time then to turn on glycolysis and the citric acid cycle. Again, ADP and NAD+ are examples of reactants during this whole process. So what we'll do next is take a look at specific steps during this whole process that can be turned on or turned off, depending on whether the cell needs more ATP or needs less ATP. So the first spot that we'll take a look at, so it turns out that one of the first few steps during glycolysis can be turned on or off depending on whether the cell has a lot or a little of ATP lying around. And so here's the specific step during glycolysis that can be regulated, that can be shut on or off. Before we take a look at that, how about if we sum up this whole slide by saying that this idea that we just talked about is called feedback inhibition. And so feedback inhibition is when a high concentration of product causes a chemical reaction to shut down. And so feedback inhibition, it's called that because the product that's formed by the reactions, it's coming back, it's feeding back in order to shut down the whole process. And so again, what we'll do next is take a look at specific steps that can be turned on or turned off. One step is one of the few steps, or one of the first few steps that occurs during glycolysis. And so here it is. So again, here's all of the steps during glycolysis. This particular step right here, we're saying can be regulated, can be turned on or off in response to how much ATP there is inside of the cell. So we'll just key in on this one particular reaction of glycolysis, focus on it, and the first thing we'll mention is the names of these two molecules that you see. So in this particular reaction, the reactant is called fructose 6-phosphate, and so that makes sense because we explained that this is a fructose molecule and its sixth carbon contains a phosphate group, so fructose 6-phosphate. This molecule here is also a fructose, but it has two phosphates on it. So to reflect that, it's called fructose 1,6-bisphosphate. So in this context, bis means 2 because it has two phosphate groups. 1,6, these numbers tell you where those phosphate groups are attached. So it's attached to the first carbon and it's attached to the sixth carbon in the molecule. Where that phosphate group comes from, so this molecule only has one phosphate, this molecule has two phosphate groups. Where that phosphate group comes from, if you remember, it comes from an ATP. So during this step, ATP comes in, drops off one of its phosphates, and then it leaves as an ADP. The name of the enzyme that's responsible for catalyzing this reaction is called phosphofructokinase. And so again, we're keying in on this one step of glycolysis because this is a step that can be turned on or off in response to the cell's needs. And so to be more specific, what we're saying here is that this enzyme can be activated or inhibited depending on the cell's needs. So depending on whether it needs more ATP or whether it needs to cut down on ATP production. So to see how this enzyme is activated or inhibited, let's take a closer peek at it. So here's an enzyme, here's a molecule of phosphofructokinase. You see here that basically the reactants, in other words the substrates for phosphofructokinase in this reaction, are fructose 6-phosphate and ATP, right? And then the products are ADP and fructose 1,6-bisphosphate. So ATP and this molecule, those are the substrates for phosphofructokinase, which means that the active site of phosphofructokinase can have those two molecules bind to it. So that's exactly what happens. Once those two substrates bind to the active site of phosphofructokinase, this enzyme can then help one of the phosphate groups be transferred from this ATP to the fructose 6-phosphate. Once the ATP has given up one of its phosphate groups, it now becomes an ADP. And so those are the things that are released as the products of the reaction: fructose 1,6-bisphosphate and ADP. So whenever this enzyme is activated, that's what it's doing. It's catalyzing that specific reaction. We're saying that there are circumstances where this enzyme can be inhibited, and so here's how it's inhibited. It turns out that it has an allosteric site, or a regulatory site, and when the inhibitor binds to that site, that prevents this enzyme from doing its job. Here's kind of the cool thing, or maybe the funny thing, is that the thing that acts as the inhibitor of this enzyme is the same thing that acts as the substrate for this enzyme. So ATP can actually also bind to the regulatory site of this enzyme and act as the enzyme's inhibitor. And so it acts as a non-competitive inhibitor, which means that once it binds to the regulatory site, it changes the shape of the active site enough so that its normal substrates would not be able to bind. Right, so ATP serves as both this enzyme's inhibitor and as one of its substrates. So ATP can either activate or inhibit this enzyme. And so kind of understanding how this works, the key to understanding how this works is to figure out the situations where ATP would bind here versus the situations when ATP would bind here. And so the thing that kind of is responsible for determining that is this. So again, ATP can bind to both the active site and to the regulatory site. It turns out, though, that the active site has a higher affinity for ATP as compared to the regulatory site. In other words, there's a lot higher chance that ATP will bind here than it will bind here. And so here we've kind of depicted that by making the slot for ATP in the active site bigger than the slot for ATP in the regulatory site. So that's not how it actually is. We just drew it bigger here and smaller here to illustrate the point that it's a lot easier for ATP to bind to the active site than it is for ATP to bind to the regulatory site. So ATP would probably have to bump into that a few times before it actually is able to make a nice fit into that regulatory site. All right, so that's kind of the key point here, is that ATP can bind to both parts of the enzyme, but it has a lot easier time binding to the active site. So ATP can bind to both the active site and the regulatory site of phosphofructokinase, but it has a harder time binding to the regulatory site. And again, the reason for that is because the regulatory site has a lower affinity for that ATP molecule. So here's kind of the consequence of that. So again, ATP can bind easier here as compared to here. So that means if there's only one or just a few ATP molecules floating around in the cell, chances are that ATP molecule will bind here instead of here, right? Because it's easier for that ATP molecule to bind here. And so when that's the case, when ATP binds here, that basically allows phosphofructokinase to do its job, which is to catalyze this step of glycolysis so that the rest of glycolysis can continue. And again, that kind of makes sense, right? Because if the cell doesn't have a lot of ATP, so if it only has a few ATPs around, then the cell would want glycolysis to happen so that it could make even more. Right? So it makes sense that, again, if there's only a few ATPs around, that you'd want to activate phosphofructokinase. On the other hand, if there is a whole mess of ATPs around, then that increases the chances that one of these could then bind to the regulatory site. Right? So if you just have a lot more of these, then you have a lot more ATPs bumping into this area of the enzyme, making it more likely that one of them will actually slip into that regulatory site. And so again, when you have that happen, when there's a lot of ATPs, chances are greater that one of them will slip into here, which means that this entire enzyme will be inhibited, which means that glycolysis will end up shutting down. At least all of the steps after this step will end up shutting down. And again, that makes sense, right? Because if the cell has a lot of ATPs already, you'd probably be interested in slowing down glycolysis, because you'd just be wasting your time doing glycolysis, making more ATP if you already have a lot inside of the cell. Right, so again, this whole process of respiration can be activated, sped up, and slowed down depending on how much product there is. For example, how much ATP there is. Here's one specific portion of respiration that can be activated or turned off. All right, so to kind of finish making the points here. So lots of ATP in the cell means that there's a greater chance that one of them will bind to the regulatory site, this site here, which then ends up shutting down glycolysis. So again, bringing it back to the big picture. Right, so you might be seeing that I'm a real big fan of the big picture. So if you were to maybe look for a place to start studying respiration, this would be a great place to start studying. So bringing it back then to the big picture, we're saying that this whole process can be slowed down. Here's one specific spot where it could be slowed down, the third reaction during glycolysis. Another spot that can be turned on or off in this whole process occurs here. So this step where pyruvate is converted to acetyl-CoA, turns out that the enzyme that catalyzes that reaction is called pyruvate dehydrogenase. And so in order for pyruvate dehydrogenase to work, it needs some reactants, it needs some substrates, and its substrates are pyruvate and actually coenzyme A. So remember that this carbon is removed and replaced by coenzyme A in order to produce acetyl-CoA. So these are the two things that go into this reaction. Here's the major thing that ends up leaving the reaction. We're highlighting pyruvate dehydrogenase because it's another enzyme that can be activated or inhibited depending on whether the cell needs more or less of the products of respiration. And so the things that end up inhibiting pyruvate dehydrogenase include acetyl-CoA, ATP, and NADH. And again, this makes sense because if you have a lot of these molecules, it's an indicator that the cell probably should shut down glycolysis and the citric acid cycle. You don't want to keep on pumping this stuff out if you already have enough of it. Examples of things that turn off pyruvate examples of things that turn on pyruvate dehydrogenase include things like coenzyme A, AMP, and NAD+. And so we've already talked about why whenever you have a lot of these particular molecules, that's an indicator that the cell is kind of hard up for ATP. So if there's a lot of this stuff, it's then a good idea to end up activating all of this so that the ATP making machinery can continue. Alright, so then to sum everything up for one last time, basically here's what we just talked about. During glycolysis and the citric acid cycle, they both release high energy electrons that are delivered to the electron transport chain where a whole bunch of ATPs are produced. So we've talked about what occurs during glycolysis, we've talked about what occurs during the citric acid cycle, we've talked about how this can be turned on or off. What we'll do in class is then talk about the electron transport chain. Right, so we'll save that kind of the punchline, the best part for last, and we'll do that in class. Taking a further step back to look at the even bigger picture, we mentioned that whenever cells make ATP, here are two options. Right, so there's there's some cells that can do respiration, and that's what we've been focusing on in this video. There are also cells that can do something called fermentation, and we talked about how that's different at the beginning. And so actually what we'll do is we'll talk about fermentation in class as well. So there's some stuff to look forward to in class.